Tilt, how are you? 1 second, I'm just setting up. I'm hoping you can actually see my thing on here. I tried something earlier for my GCSE live session, and I'm hoping I'm good. Wow, I can see, yay, it is working. That's what I like to hear. It's exactly what I wanted you to say. Thank you. That confirms what I wanted. Right. Which also means that I'm here and there at the same time, which is good, because that will go up there. I'm going to close these ones down just now whilst I leave only that one. That I am good, thank you. I am good. I am literally just working away. Alex, good to see you. Haven't seen you in a while. How are you? Hi, are you early? Buffy, I've only just started and come live now. So you're not early, you're right on time. I was a little bit late, to be honest. How are we all feeling though? How are we doing? What have we been learning and what topics are we struggling with? Have we got any specific questions? Or anything you want me to go over? Hopefully, for those of you who are just joining me, I have been doing this for quite a while now, and I keep playing with things. So this is the first time I've managed to get OBS to actually do it in two places at once. Woohoo! Exciting. Right, Alex, what are you saying? Great to see you. I'm good, thanks. How are you? I'm very good. I am very good. Graphs were hard for me to grasp on your mock paper. What sorts of things is it that you struggle with graphs? Is it the interpretation? Is it the drawing? What is it? We just finished beta cells and today we did alpha cells. We don't have to know the processes, but diabetes. Yeah, so the beta cells, you do need to know the process of how the beta cell is going to secrete. So you should know that one, the interpretation. Okay, let me see if I can grab, excuse me, just a random question then. Let me see if I can find a random question for us to look at, which covered, yes, we learned, I love the beta cell. I love that information. Sorry, I've just went in the house really quickly, wolfed down some food, and consider I'm freezing, an ice lolly as well. And I am going to just quickly bring in a question. Right. Yeah, you don't need to know for the alpha. You just need to know about the actions of glucagon. It's the insulin secretion that they want you to know. Conventional versus intensive. Yeah, they did a long question on that one actually, Alex, where in the past they wanted you to compare it was a 6 marker to do with old treatment, new treatment, and the potential of treatment in the future as well, which was like obviously stem cells. Right, we want graphs. We want some graphs. Let's have a look. Let me see if I can quickly find you a random graph we can look at and interpret together. Actually, do you know what? I just did a question with my group tuition sessions. Let me bring that one in, because that's quite a good one. I do like it because it covers so many different areas. Miss, we had our test in transport and cell membranes. Oh, well done. You've done it. That's the key thing, that you've actually done it. So now it's about how you move on from that, and what you figure out what you don't know. So the key thing now is the stuff that you didn't do so well on, identifying what it was that you struggled with and how can you move forward to improve for next time. So don't take it as a knock if you get the things wrong. The key thing is to use that as your guide on what you should be revising. Right, am I happy with that? Yes. And then let me bring this one in as well. 1 second. Bear with me, guys, as I bring in some stuff. Nope, do not mean that and that. Okay, so what I'm going to look at then is we're going to start by looking at a practical to do with respiration. So it's important that when we look at respiration, that we are looking at the topic areas of all of it together, not just one small part of it. Okay, so they could ask lots of different questions to do with respiration. So let's imagine that I have got a yeast stock solution which has been made up from 10 grams of yeast that's been put into 250 centimeters cubed and has a pH buffer of 5. So I know that they have controlled the mass of the yeast going into my stock solution. I know that they have controlled the mass of the sugars that are going into the solution. And then what they're going to do is, if they were to take 25 centimeters cubed of this and 25 centimeters cubed of this, and they were to place them into the new sample. So this is 25 centimeters cubed plus 25 centimeters cubed. And they are putting it in a water bath, which is 35 degrees C. And then they're going to measure the volume. So they're going to measure the volume of gas that's collected. So, excuse me. What they could do is they could give you a question which outlines this in the STEM, where it says this is the method that the students completed. So what I would say whenever you get a practical situation is to think about, what have they told me? What do they want me to give them because of what they've hinted at in the question? So straight away, any practical, you should be talking about your IV, DV, CV, and you should be looking at what they are going to actually be controlling. So we've got the water bath here, which is at 35 degrees C, so we're controlling our temperature. Why is that important? What's temperature got to do with it? Guys, thank you for all the likes, by the way. I'm not missing this, I can see it, and I do really appreciate it. So the temperature, why is that important? Now, anytime I see anything to do with temperature, straight away, temperature is my trigger word. And the next thing that I want to write is kinetic energy. So I want to write about my kinetic energy. So I've got my temperature. Is it a low temperature and therefore there's less kinetic energy? Is it a high temperature and therefore there is a high kinetic energy? What is it that I want to talk about? When I talk about temperature, I need to be thinking about that kinetic energy. So I've got a water bath, I've got it at 35 degrees C, therefore I am controlling my temperature. I was told that we had a pH buffer of 5, so therefore I am controlling my pH. Why? Well, this practical that we're going to look at is to do with respiration. And what do we know about respiration? Random question. Do you record during FM? What does FM mean? Do I record during these sessions? These are currently on my YouTube channel. Recommend. Do I recommend it? What a completely opposite way. Do I recommend, honestly, I am awful at abbreviation. I can work things out. Do I recommend doing further maths? If you're good at maths, yes. No, don't apologize. It's really funny. Yes, if you're good at maths. Do I recommend me doing further maths? Absolutely not. I'd rather run for the hills. I hate maths. I really do not like it. Yeah, it's really not, it's not my cup of tea. So for me, no. If it's something you want to do later on and it's something that's going to help you, then yes, it is good. Um, it's not going to harm you, but would I say you need it to get into meds? You don't desperately need it, but it will help you. Alex, that's amazing news about your interview for med. That's really awesome. Well done. You must be so proud of yourself. The next thing is about the interview stage. That's the hard part. Actually, it's more fun. Treat it like you are summing them up if you want them to be the place that you go to as well. That's the key thing with interviews. Right, back to this. The next thing I'm going to say to you, whenever you're told any information, this is the bit that students get hung up on. They forget that they need to go back to module 2. So you were told that you have a glucose solution. You are told that you have a maltose solution. You are told that you have a lactose solution. You are told that you have a sucrose solution. Also, was the person asking if it's, yes, they were saying, is it useful to do further maths, Alex? Alex says, in response, Alex says, no, it does not help one bit for med. So, in terms of time, especially if you're applying for med. Yeah, Alex is saying no. He's currently year 13 and he's got an interview for med on the 9th of December. So he's saying no. Other people will say yes. It just depends where your strengths lie and what you want to do, I would say. But I'd be running away. It's not for me. That's why I'm a teacher, because I found my niche. I found what I like to do. I want to help students. I want to do what I do here. And so, yeah. I'll have to think, because I really, well, this is the thing. If you enjoy maths, then if it's not going to, it's hard. Just to be clear, I've had students who've done maths, further maths, chemistry and biology, and they have struggled with the exams on the further maths. So it isn't an easy task to do. Yeah, don't do it. Alex is saying, and that's a really important thing, Alex, that he's saying if you enjoy it, that's fair, but don't feel like you need to do it or be obliged to do it, because actually that's not what's going to help you. You've got to study whatever you've chosen for, if you're in year 13, the next scary prospect, but 6 months. And so, but if you're currently year 12, which I'm assuming you might be, then it's something you're going to have to do for the next year and a half, and it's not easy. So Tildes does biochemistry for med, but then she does French and Spanish, as she loves them too. And that's the key thing. It's doing what you love alongside the stuff that will support you and help you. They want to see you as a well-rounded person. Sometimes if you narrow it down too much, you do maths and further maths, and yes, that will give you a little bit. However, it's not something that's going to give you a step up over other people, I would say. It depends on the university, really. Skills a bit weird and forces us to do 4. Yeah. That's not a bad thing, actually, to be fair, Darrow, in the fact that if you do 4, you can figure out what you don't like. You'd be surprised how many students. So when I taught the IB and was head of biology, people were constantly shifting, and we normally would let them shift up until probably the end of the first term, no further than the first term in year 12, because you're figuring it out and you've got time to pick up what you hadn't done already. But yeah, it's about having that balance between it all. That's the thing I would say, is making sure you don't just go solely down the academic route because you think that will get you a step up, because it won't. Anyway, back to this, because you were asking, if you're still in here, about graphs. Oh, where are you? I feel really bad. I didn't notice your name. I'm sorry. Buffy. If you're still here, Buffy, I apologise that I went on a bit of a detour. Yes, these videos are on my YouTube channel as well as here. How hard do you think IB biology compared to A-level biology? Which one? Oh, I love IB. I love IB. I taught IB for 11 years when I went to be lead teacher before I became director of student progress, before I gone on to do this. But I love the IB. I just love it. The reason I love it is because it's first year university experience. I would say, oh, thank you. Yeah, definitely tap the screen for support. That'd be really lovely. I would definitely say they just have different ways. They're all level 3 qualifications. So as I said to someone before, the difference between whether it's AQA, OCR, IB, whether it's EdXL, there is no real difference because you're getting a level 3 qualification. But what I love about the IB is that it works for the way that my brain works, in the sense that it gets you to tell the story. Although I hate paper 2, data analysis questions, they're horrible for all students. But I love the multiple choice paper 1, I love the long response of paper 2, and then the paper 3, which has changed slightly since I last taught it because they've removed the options. But I love the IB, and I would say they're on par because they're all level 3 qualifications. Oh no, I love the OCR and multiple choice question. I love multiple choice questions anyway. I mean, that basically got me through my first year of university. The OCR and multiple choice questions are difficult because they make you second guess. And that's the problem. But if you eliminate and you spend time learning how to eliminate, you'll actually thrive at them. And you'll be able to see where questions could go in future years, because what they tend to do is if they've asked a topic quite a lot in long response questions, then the following year or 2 years time they will ask them as multiple choice questions, because that way they can cover the whole spec that way. So there's a little tidbit for you on how they do it. If you do any analysis on the papers, you will actually see, oh, they asked that in the multiple choice there, so the next year that will be a 3 to 4 marker, or they'll combine those together and that will push that one a bit further. Right, back to this. So I've got my different saccharides here. So it's really important that you note that you have got glucose, which is a monosaccharide, our maltose, which is a disaccharide, lactose, which is a disaccharide, and sucrose, which is a disaccharide. So we have got 3 disaccharides and 1 monosaccharide. The other thing for us to note, just because we're doing a bit of a recap and refreshing, not only are they disaccharides and monosaccharide, but we also have a non-reducing sugar and reducing sugars. And so therefore, because we have our non-reducing sugar and our reducing sugars, they could ask a question about how you would do testing for whether or not you have reducing or non-reducing sugars present. So do you know which test to use? Do you know what colors, if it's negative and if it's positive, could you give that? What they might do, which is what I wanted to show for Buffy if you're still here, is a graph of a set of results. Now, I love this question because it's such a nice question for really assessing 2 things. 1, can students interpret the graph? And 2, are they actually reading the questions and understanding what's been asked of you? So let's start with what are they telling us with this question. So I don't know what I've got in my eye. It's not eyelash, but it's really bugging me. The first thing we need to know, I can't actually see anything now. There we go. The student investigated the effects of different sugars on the respiration rate in yeast. So we've got different sugars, and we can see that they are the different sugars because they've actually told us they're sugars. We've got maltose. So this is my maltose. And this is the other thing I would suggest you do in the exam. If you have access to highlighters, take highlighters in with you. Highlight the different things that you're looking at, because if you highlight them, it makes them more visible so that you can then interpret them in a better way. Whereas if you are just looking at them and looking at a black and white graph, it's really tough to then distinguish between, oh, back again, whatever it is. This is a funny one, actually. This is a blink response because there's a stimulus, which is being detected by my receptors, which is pressure, mechanoreceptors dealing with pressure, something is irritating my eye. And so therefore that is sending an impulse via sensory neuron to come back via motor neuron to cause the muscles to contract, to cause me to blink, to try and get it out. It's a reflex. I'm not thinking about blinking. It's happening because something is in my eyelid and it's damaging my eye, or potentially damaging my eye. So my body's responding in such a way to make it protect me. There you go. There's biology in action. Yeah, it's the reflex. I love it. Oh, I love biology when you can bring it into real context, but it's not nice that my eye is really hurting. Anyway, back to this. We've got these different sugars, respiration rate. Guys, if you've just come across my channel as well and you have no idea who I am or what I'm doing, welcome. I'm Christine. I am one, neurodivergent, if you haven't seen that already, the way I am chatting and responding. Two, I also love biology and I want to help you guys learn as much as you can. So please do keep liking, sharing and watching and buying my revision guides. I was super excited. I put my revision guides down to 99p each this weekend and I haven't actually put them up yet. So you can get them at a very bargained price of 99p for each one individually. No worries, Alex, no worries. Oh, bless you. Have a good one and good luck on the 9th. If I don't see you before then on your interview, I look forward to hearing how it's gone. You're welcome. Daryl, yeah, for you, if you're 12, you want the nucleic acids nucleotide one and the biodiversity one. And even though, and the transport and animal one and the transport and plant one, actually, the transport and plant one goes into photosynthesis that you won't need yet until year 13, but it's got that and plant response all together in one because it tells a story. So that should definitely be helpful. Oh, the nucleic acid nucleotide one then will be really helpful on that one. Because I've got some really lovely diagrams and it goes through all the content that you need for the topic area. So definitely check that one out. Anyway, back to this one. What have we got? We've got these different saccharides, different sugars, and we're looking at the respiration rate. So for year 13 students, you should be looking at this and saying, oh, respiration, volume of gas collected, they're there for collecting carbon dioxide. So if they're collecting carbon dioxide, where is the carbon dioxide being produced? Which parts of respiration? So if I'm looking at respiration, I therefore want to be talking about, is it in glycolysis? Is it in the link reaction? Is it in the Krebs cycle? Or is it in oxidative phosphorylation? Now, right now we are not doing the exam. So right now we are just doing some revision. So if I'm looking at a graph and I've got the time, I would be making sure that I'm going through this kind of content to say, right, oh, carbon dioxide concentration, because they haven't asked it in this question, but they might give this graph again and ask it in a different question. So that's one of the things to be aware of. They do reuse some graphs, but ask them in different ways. And students go, oh, I recognize that graph. I've memorized that mark scheme. I know what they're going to ask me, and they never do. So it's important that you note what it is they've asked you, that you note what it is that you need to talk about. So if it were asking about where does this gas come from, you should know that decarboxylation occurs during only the link reaction and the Krebs cycle. It's not what the question's asking. However, that could be one of the questions. What this question is actually asking is for you to interpret the graph. So you have been told that you have students who've drawn a conclusion based on what they can see. So they have said that the rates of respiration, now remember, rate of respiration is quantity over time. So if I'm looking at working out the rate, I ideally would want to be drawing a tangent line to figure out my rate of respiration for each of the ones that they have named in the example. So what have they named? They have gone with glucose, they have gone with maltose, and they have gone with sucrose. And they have said that the rate of respiration in each of these are very similar. Well, if I were to draw, as I have done, my tangent and calculate that, if I bring that over to the next one, oh, that's different. That's not got a steeper gradient. So therefore, that would be something you want to talk about. Hi, Daryl, thank you for coming over. So when you look at graphs like this, you need to be thinking about, do you know how to work out the gradient and to work out the rate? So do you know to use your ruler? Sounds ridiculous saying this, but so many students don't use rulers. I didn't when I just drew that. Is that a good way of doing it? Absolutely not. So make sure in the exam you get in, whether you're doing it as a practice exam or whether you're doing it as the real thing, you have practiced using your calculator, drawing tangents, things like that, because that's going to be the difference between you doing something faster or not. So if you have not done it in a long time or practiced it and you try and do it firsthand in the exam, you may make mistakes. So try and get into a habit of doing things from the way you will do it in the actual exam. So in this case, I'm trying to use my ruler on my slide. So if I bring that across, bring that down, let's get rid of you. So then I can work out my tangent. Could we please cover the cell cycle for mitosis? Yes, Tills, let me just finish on this graph and then I'll come back to that one. So is it very similar? Well, no, it's not very similar, but actually they do follow the same trend. So you want to be talking about what you can see. The slopes are different. So that is a 4. No, it's not. That's an against. So evaluate, I want to say a 4 and I want to say an against. I want to be arguing both sides. Anytime I see evaluate, always argue for and against. Yes, this is true because. No, this is not true because. And that is number 1. That's the first one you're looking at. And then you want to be looking at number 2. And that number 2, it's about the disaccharides. Do you know which ones are disaccharides? Do you know which ones are monosaccharides? Do you know the difference? So in glycolysis, which one are we going to be using? Obviously, our respiratory substrate of choice would be glucose. And therefore, if that's our respiratory substrate of choice, what have we got with maltose? Maltose is our alpha glucose with another alpha glucose. And sucrose, we're talking about a glucose and a fructose. And our lactose is a glucose and galactose. So we know that maltose, sucrose, and lactose are all disaccharides. They all have glycosidic bonds, and therefore they are disaccharides. Oh, Daryl, thank you so much for buying all the study, guys. That's really lovely. Thank you. I really appreciate that. I'll send an email later to say thank you. But I do really appreciate you purchasing them, and I hope you find them really useful and you like them. Definitely check them all out. Hi. And if you have any questions, please do ask away. Right, Tills, you wanted to look at some of the mitosis content, right? Oh, what's the answer? So the answer would be for number one, and it's about interpretation of how you answer that. The rate of reaction with glucose, maltose, sucrose are very similar. Yes, they follow the same trend. So I'm saying yes, because they are following the same trend. However, I would want to be talking about the against in the fact that they are the different slope sides. For number two, the yeast could not hydrolyze disaccharides. Well, that's not true because maltose and sucrose are both disaccharides and they were hydrolyzed down because they did produce carbon dioxide, which therefore means that they were used to produce the pyruvate for the link reaction and the acetyl coenzyme for the Krebs cycle for the decarboxylation to occur. And so therefore I can say that's not true because of that. However, again, on the opposite side of things, the lactose does not increase at all. So the lactose isn't actually able to be hydrolyzed. And why is the lactose not able to be hydrolyzed? Well, this is where they could bring in the lacoperon, if any of you have done cellular control, in that the lacoperon is useful because the lacoperon for E. coli allows for the E. coli to actually be able to produce the lactose permease, to produce the beta-galactosidase, to be able to produce the lactose. So the fact that they're not able to hydrolyze the lactose means that they probably don't have the enzyme lactase. So I'm making a conclusion on this, but it's not true that all yeast could not hydrolyze the disaccharides because some of the disaccharides were hydrolyzed. So it's about that for and against. Always trying to find what's a for answer and what's an against answer. I have a B1 exam tomorrow. What can I do to be prepared the most? I would say if your exam's tomorrow, you should have been doing lots of revision right up to this point. I would say try to continue to go over some flashcards. Not even joking, those notes look, thank you. I feel like most of the stuff out there for OCR A is outdated, especially. Yes, I don't know about other people's stuff out there, but I specifically built, and this is why I'm still building them. These study guides have taken me a long time to do because I want to make sure that one, they've got enough information for you. The extent of the teaching for nucleotides, nucleic acids, doing DNA. What? You did a DNA dance in your lesson, and that's how they. Was that the helicase to break the hydrogen bonds, and then the replication forks that were then your polymerase? Oh, it was so bad. Do you know what the funny thing is? When I'm in lessons, that is literally what I would do with the songs and that. My study guides are 99p. Each one is 99p, and you're very lucky because the transport in animals and exchange one that I've just done, and the two brand new ones, I have done them more in the way that I teach. I was doing the individual guides for each of the topic areas, and then I realized that's not what my strength is. My strength is, one, my diagrams are great, if I do blow my own trumpet here. But I spent a long time doing Adobe Illustrator as well, learning how to make the diagrams and stuff. Also I teach it in a story, and I learn things in stories. And so I've told the story that it should interconnect and make sense. My study guides and the exam board that I work on mainly is OCR A. So all my videos that you're watching are on OCR A, but there is a massive overlap. My one-to-ones that I do for AQA, I adapt the resources. So I actually have just done what did I just do? The centrifugation. I just did a little diagram on that for one of my students. and I was doing viruses today for one of my students as well. So I am adapting them. The format and layout is so clean. Oh, thank you. That is what I wanted it to be. I wanted it to be, there may be some slight bits that I need to go back and double check with positioning, but I was like, I just have to stop being such a perfectionist. But they should be as clean as they can be. Do you think I can revise all of biological molecules and cells in a week? Oh yeah, absolutely. That's an easy one. Biological molecules and cells is the easy part of biology. Sorry if that's going to burst your bubble, Aisha. I really don't want to make it seem awful, but it is hard. And it gets harder. But if you can nail those two, you're in a very good starting point. Tildes, I've not forgotten that you're wanting to look at mitotic division. Give me 1 second. I'm just getting up the resources whilst also answering these questions. Hardest possible question on a protein nucleic acids test. Hardest question, I would say, I don't know. I actually find that stuff easy. Now, because I've done it so many times. Hardest question, Darrow, you're year 12, so the stuff that's to come later is much harder. But I'd say most students fail on transcription, translation, so protein synthesis, trying to put that together. They're happy with bringing in like the RER with the Golgi, etc., but they really struggle with the tRNA and what happens at the ribosomes. And bring in the biological molecule side of things, where you've got two amino acids and how a peptide bond would be formed between the two. The DNA replication side of it, they've asked one question that was really tough, bringing in the mission style side of things, but I don't think it's that complicated if you look at the question and you describe what you could see. Yeah, basically memorize your key trigger words of like the processes, breaking the hydrogen bonds, the complementary base pairs, etc. Things like that. They're your important ones. But primers and oxidizers or whatever are irrelevant, right? They're not irrelevant. So I've kept them in there. So the reason they're not irrelevant, Darrow, is because when you get to year 13 and you start doing things like polymerase chain reaction, and you start looking at how you join together your gene and your plasmid, you need to know about ligase, you need to know about the primers that need to go in. So it's not that it's irrelevant, it's that it needs to be there for you to see where it's coming. We love Okazaki fragments. We love it. And all the different keywords. Like I love saying pseudopodias when I talk about phagocytosis, whereas so many people are like, you don't need to know that. I'm like, yeah, I know, but it's such a cool word. I just like saying it. So hold on, Tails, what did you say? Have you heard of the new organelle, the hemifusum? It basically manages cellular recycling and helps to diagnose. Oh, that's really cool. Do you know, it's a real, I've been so hyperfixated and focused on what I've been doing lately. I've not really opened up to the other things that have been going on, and I need to. It's really bad. I have been so blinkered lately. We've got school play rehearsals going on. We had gymnastics, so I dropped the girls off to gymnastics. Whilst they're in gymnastics, I do a little bit of prepping if I can, but today they were just doing stuff that I had to watch. And then after that, it's then straight on to, I do a live for GCSE at 6:30. Then I do a group tuition. Then I come back on and do this here. So yeah, it's sometimes I don't get a chance to just stop and breathe, but it's all good. Right, I am coming with this knowledge on mitosis for you. Yeah, so pretty much everything is really fascinating. Everything goes to the Golgi to be modified. And as long as you remember that the Golgi apparatus is where you're going to modify things. So when you get to module 6 and we start talking about the modification post-translational regulation, then what we're talking about is the adding together of the different things like the carbohydrate being added to the protein to make a glycoprotein, for example. And then remembering that the fact that you are going to have the Golgi vesicle, which is going to fuse, means that that can become part of the membrane structure. I just love how it all interlinks. That's one of the like best things about biology, is that it all interlinks. Right, Tildes, for you, we're going to do the cell cycle and go from there. Okay, so you obviously know interphase, G1, you've got your different things that need to be checked. Now, this is again where we need to link together our topic knowledge from module 3, let's say. So interphase, we know that G1 is about our cell growth. Well, when our cells start to grow, so let me just, come up here. When we have a cell, so I'm just going to draw it as a cube, and that cell starts to grow. So if I take that, no, 1 second, let me try again. Here we go. If I take that cell, let's put it here, and I make that cell now grow and become bigger. There we go. We have two cells. We've got a cell that's gone from one size to now being bigger. So if I had a cell that was 1 by 1, and then I've now got one that's 10 by 10, what we can see is this cell is expanding, it's getting bigger in all different directions. So therefore, if I take my small cube and I take my big cube and I separate them, then what I should notice is that my surface area to volume ratio is changing. So my surface area on this one, which is a 1 by 1, would give me a 6 to 1. Whereas when I look at the 10 by 10, I then get a 0.6 to 1 surface area to volume ratio. Yep, it's definitely A-level biology. We got to love A-level. A-level biology is awesome, but it's also hard. And everyone who's like, oh, pick A-level biology because it's so easy. No, it's not. It's tough. There's a lot to learn, and you have to really know your content, especially if you are going up to aim for that A-star grade. And that's the one thing, like if you stop doing it, even for the summer, anything like that, you'll be surprised how much you will forget. Like me as a teacher, I will have the summer holidays and be like, I don't even remember what I was teaching. I don't even remember what I was teaching like an hour ago, because my brain is just so full of all the other things that are going on. And so you have to repeat, you have to keep going, you have to refresh and go back. So those students who think, oh yeah, it's fine, I'll do it later, then actually they're the ones who miss out on grades because they haven't taken the time. And that's an interesting one. So many people actually do think that the content is good, but their application is hard. And I'm not picking up on you to be mean, but what I would say to you is a lot of students feel that they know the content, but they only superficially know the content. And I mean that in the nicest possible way, in the fact that if I were to question you, which is what I do when I do my one-to-ones with students, I'll say to them, let's do this topic area. How do you feel about that? And you're like, yeah, I'm really good on that. And then I'll ask them a question. They'll go, I don't know how to answer that. And I'm like, well, actually, that's because you don't know that content as much as you thought you did. And that's the bit where you struggle with the application, is potentially you've not delved into the content deep enough to be sure that you really know it. Have you checked you've got no misconceptions? Are you able to expand on why something is happening? Or are you just able to regurgitate what you've read in the textbook? And that would be the sticky point. The mark schemes are brittle and the mark schemes change. This is what's frustrating for you. You feel like you have just got to grips with one mark scheme and then it completely changes. And then you're like, well, hold on a minute. You accepted it this year. Why are you not accepting it that year? And that is because those mark schemes are specific to that year of those who sat that test. the examiners that marked it that year. The next year, the next set of examiners that come together agree or disagree what's allowed in that mark scheme. And so that's where you have to be aware. Where can I make sure I know everything in detail? That would come as that comes from doing lots and lots of practice papers. And when I say do practice papers, be harsh on your marking. If you are looking at the mark schemes and you are marking your own ones, please be very harsh on yourself. I nitpick away, and some of my students are like, oh, but Christine, I said this. And I'm like, no, you didn't. You implied it. So there's a difference between you implying something and the person who's going to mark it actually seeing your answer, knowing what it is that you know, and giving you the grades for it. We do not see you. We do not talk to you. We do not have any connection with you whatsoever. So we're not going to give you those marks. Just because you've implied something does not mean that you have made it explicit to us, an examiner, that you know it. So that would be one of the other things I say that is a downfall. It's good. What have you said? I have a biology test on biological molecules and cell structure. Have you covered the... Yes, I have. If you go to... It's good. If you go to cell structure 2.1.1, biological molecules 2.1.2, go to my playlist and look at the videos that I've got there. Also, what about people like Amoeba Sisters, just visual animations of everything in terms of actual understanding of the process, then learn these? Yes, Amoeba Sisters is really good. I would love to be able to make Amoeba Sister videos, quality. They're really good and I like what they do. One of them is actually science trained. The other one is the animator, and I'm kind of trying to fumble my way through it all. So yeah, I would definitely agree with Amoeba Sisters. I haven't seen any of their more recent one. I don't know how much detail they're going into with A levels, but I used to use Amoeba Sister with my GCSE all the time. But I definitely do think that is good. The visual side of things helps massively. Is STEM up flashcards good? I don't know. I can't tell you whether they're good or not because I haven't seen them. I don't use them. I would say flashcards are good as a way of revising. However, be cautious that you don't rely on the fact that someone else has put the thought process into narrowing down the content into keywords. So flashcards work because what they do is they have that trigger word. That's what a flashcard does. And that's why I always use the words like trigger words. What are your trigger words for a topic area? So when you make your own flashcards, when you read content, when you watch a video, when you do whatever revision technique that you have got and that you do, when you're doing that, you are thinking through all the things to do with that topic area. If you're then having to summarize that down into 5, maybe 7, 8 to push keywords, okay? So this is one of the activities I get some of my students to do. If they really struggle making their own notes, I will say, right, you're going to look at this topic area. You are going to bullet point it down, have your trigger words. I want no more than 8 bullet points, maximum a topic area. And then I'm going to ask you those words and you then need to make them into sentences. You need to expand on them. You'd be surprised how many students will just rewrite every word. They'll have like 1,000,000 bullet points with every single word that is relating to that topic area. That is a waste of time. That is not a useful way of you revising the content. A useful way of you revising the content is reading the textbook, reading the revision guide, reading the study guide, watching a video, turning that into your own understanding, your own keywords, your own way of writing things. There are 20,000 of you who sit this exam every year, okay? 20,000 of you. You are not writing it the same way. There are keywords you need to use, absolutely 100%. But if you can't use the keyword or you don't know the keyword, describe what's happening. So find that way to make it the description and then use that and expand on that in the exam. And that's where the reason I did this, and I've gone a bit around the ways with, the reason I've said it this way is because people buy these flashcards They then don't know how to use them. And so what they're doing is they're buying them and not utilizing the benefits they would get from them in the right way. So I can't say whether they're good or not. I don't know because I haven't seen them. However, flashcards are good if you use them correctly. You making your own flashcards is better but very time consuming. So it's that, which one do I do? Do I buy them and use them in the right way, or do I make my own but it takes me a long time? That would be where it's a bit of a sticky point. Again, Cognito, I don't know. I haven't used any of this stuff. I've been so busy building my own. Basically, when I came to start teaching OCR again 3 years ago, I hadn't taught it for a number of years. I'd done AQA, I'd done the IB, I'd been doing all of them for such a long time that when I switched to OCR, I was like, oh, I've got to quickly learn this content. Where can I go to look for the content? And there was nowhere. Where can I direct my students if I don't have the time to be able to teach it to the level that I want to teach it to? There was nothing there. I started doing TikTok because I was promoting AIQchat.com, which was my revision platform that I built with my partner. And so I was promoting that, and my students were like, oh, go on TikTok. I was like, what is TikTok? And that was like 3 years ago. And then when I went live for the first time, just before the 2023 exams, the students on there were like, oh, do a YouTube channel. So I've learned Adobe Illustrator, I've learned TikTok, I've learned YouTube. I've been learning so many things on here. I haven't looked at all the resources that are out there, and so I couldn't comment. I don't want to say anything good because I don't know. They might not be very good. I don't want to say anything bad because I don't know. They might not be that bad. I don't know. I'd obviously recommend my revision notes, but that's because I've built them and I've done that. But that would be for someone who, one, likes the diagrams they see in my videos, and also who finds that through storytelling and going sometimes a little bit further into the knowledge helps them to understand it. I hate when I don't understand something, so I have to do research. Kerbudo is again another one that's fine. I have actually used a lot of Kerbudo stuff in the past because the previous private school I was working at had Kerbudo, and I looked at that. So yeah, that's not too bad, that one. If my own flashcards will work either. Oh, should I put command words? Yes, absolutely. Your flashcards should mainly be the command words and the trigger words for you on that topic area. Absolutely, 100%. I got a B in biological knowledge by using my own flashcards, but then I realised I need to do more exam questions. Yeah, using the two together, that's really, really good. Do your flashcards first, then the exam questions, and relearn flashcards. I wouldn't just say relearn flashcards. The other thing I would say is to add into that, because that's a really good bit of advice, but I would caveat that with do your flashcards, then do the exam questions, then look at your flashcards with the exam questions after you've marked it. See if there's any gaps, any bits that were in the mark scheme that you didn't add in your flashcards that you think are really important that should be there now. Because when you first do your flashcards, you add extra bits, and it's about honing it down to what you need. Yes. Tips for paper one mocks coming up soon. Do I have any tips for paper one mocks? Watch my live video. I'm not joking on this one, actually. If I can give you any prep, I'd say watch my live video that I did. Would have been the day before the first exam, so the 4th? The 4th of June. About the 4th of June on here, I went live for about an hour and a half before the real exam. Did my predictions of what I thought was going to come up, and everybody came out of the exam basically being like, oh my gosh, Christine, you got it all right. So I would definitely say go and check that one. A product manufactured using microorganisms and single cell protein. Describe how a protein would be synthesized in the cell of a single-celled fungus. Yeah, so describe, because it's a describe command word, how a protein would be synthesized. It's being synthesized. The fact that they're saying it's a single-celled fungus means it's eukaryotic. you should therefore know that you've got your DNA, which is your linear DNA, which is going to be separated by your helicase. You've got your RNA polymerase, your free nucleotides, the transcription. Then you need to talk about the translation. So I would be bringing in that it's an ATS ribosome for the translation part of it. To make my own cards, but people say Anki is good. Would you recommend? Again, I've never used any of this, but I do hear Anki is a place that people can use it. So it's because I say use which word. And I think that's really good advice. You've got to figure out what works for you. Just because it works for someone else doesn't mean it's necessarily going to work for you. OCR A is the exam board. Make sure you actively pose it. It's definitely not about memorization. Tilde, I will finish this and then I will stop for tonight. Sorry, I have gone a bit of a detour on this one. Okay, so G1, the growth, we were talking about the surface area to volume ratio changing. So when the cell gets too big, the cell will then need to go through to the next stage, which is the synthesis stage. So what you need to understand is before it can move through for DNA replication to occur, you need to understand that the cell needs to be the right size, it needs to have the right nutrients, the right growth factors, i.e. it needs the enzymes to be produced so that it can then go on and do the next stage. And if there's any DNA damage that has occurred, that needs to be repaired, because if it's not repaired, that damage is going to be replicated too. So that needs to all be checked before it can be moved on. So this here is my G1 checkpoint. If we go check, 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 yep, 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 yep. Now we can move on to S phase, and this is where your semi-conservative replication occurs. Moving through to G2, your cell growth again. Again, we've got another checkpoint. So here's my checkpoint at the end of G2, double checking the size, double checking that replication has occurred in the right way, double checking that the DNA isn't damaged before we move through to mitosis. So mitosis obviously is necessary for this growth, tissue repair, and asexual reproduction. So if we're talking about plants, animals, or fungi. Now, mitosis is split up into those different parts. So somebody I noticed said earlier about IPMAT. So I always go with PMAT, prophase, metaphase, anaphase, telophase. I never include the interphase because interphase automatically are break up into G1, SG2. So when you have the mitosis, which has your prophase, metaphase, anaphase, telophase, note that there is another checkpoint. There is another checkpoint here before it will continue. So for those of you who have done the cell control part of year 13, you should know that there are genes which are going to stimulate cell division, and there are genes that are going to suppress cell division. So cell division is a controlled process. So we want cells to divide when they are needed, but we don't want it to be an uncontrolled cell division because that's how we end up with tumors. So therefore, obviously our tumor suppressor genes are going to reduce the cell division. So we have these very specific cyclins that need to be at the right concentration before it will move to the next phase. Now, you don't need to know any of that part of it, but just be aware they may give you a stem of a question that tells you some information and you're pulling out of it. If you go to my lives, it should be in there somewhere in the list of videos from lives. I don't know how it shows them to you, but it should be in the, if you go to my channel, scroll along to it, it says live, click in there, and you want, I think it may have been called paper 1 prep, something like that. You are very welcome. Okay, so what's happening during S phase? DNA replication. So DNA replication. So here you can see I have my chromosomes, and I've used Play-Doh in this one. So I got my Play-Doh, the kids had Play-Doh out and I was playing with them. So I made chromosomes, obviously. And so my green parts here are my centromeres. So that is a chromosome, that is a chromosome. These ones, you can see the green part is my centromere here. Up the top are my telomeres, and at the bottom, so top and bottom. And then I've represented different alleles of the same gene with my random colors. There's a blue and a red, and there is an orange and a yellow. So when DNA replication occurs, whether we're talking about mitosis or meiosis, DNA replication has to occur first. So the DNA is replicated, and so now I have chromosomes that are made up from sister chromatids. So these chromosomes now are chromosomes made up of sister chromatids. So during prophase, your nuclear envelope and nucleolus disappears. Your chromosomes are going to condense, and that's where they supercoil. And then your centrioles are going to move to the poles. And they are going to start to produce the spindle fibers. Now, spindle fibers are part of your cytoskeleton. And those spindle fibers are rope-like proteins that are going to come out, and they're going to attach to those centromeres. So the centrioles have moved and the chromosomes have condensed. And that's why we draw them as X shapes, because we've seen them where they've been condensed right down. And I'm looking to see if I've got something that I can use. I will be able to use that now. There we go. I'm going to randomly use this. So what you get when you look at your DNA, what you should know is that eukaryotic chromosomes are, their DNA is associated with the histone protein. So my hand, imagine my hand is a histone protein. What I have is that the DNA is wrapped around the histone protein twice. It's called a nucleosome. So right now, both hands are two histone proteins with the DNA wrapped around them twice. Now, when we're in prophase, what's going to happen is these are going to supercoil and condense. And what that means is that they're coming together in a much smaller area. So they are supercoiling, they are condensing into the chromosomes, which are now visible. And so what you can see is the difference between this one to this cell is you can start to see the condensing that's going on. It's not fully condensed yet, it's only started to condense, but we are seeing it start to condense. And when we look at it, we see these different cells. You can clearly see the nucleolus in most of these cells. And then you see this one here. And this one here is showing you mitosis. Let's try that again. Metaphase of mitosis. Because what we have is that they have lined up at the metaphase plate. So we have the fact that the chromosomes have lined up at the equator in the middle, and you see them because they're lining up. Now, whenever I do the diagrams, whenever I'm showing the chromosomes, I've shown them in a way where they're really nice and straight and on top of each other. Well, they never sit like that. They sit in lots of different ways. And so therefore, you see them kind of poking out, but the majority of it is at the metaphase plate. Crazy, I don't have time left. I'm just finishing this off for Tills if she's still here. But if you go to my channel and you look at, hold on, you're doing immunity 4 point, it's not 4.1.1, what is it? Module 4, maybe it's 4.1.1. You're looking for communicable diseases. Hold on, give me 1 second. It's definitely module 4. It's whether it's 4.1.1 or not. Which one is it you want? You want... Oh, it is 4.1.1. Go to my playlist and go to 4.1.1 and all my videos on immunity are there. And so you can watch whichever one you need, which should cover you with the content that you need. You're very welcome. Okay, so metaphase, we have them lining up at the equator. Anaphase, so we can see anaphase down here. And the reason we can see anaphase down here is because you can see these V-like shapes of the chromosomes. Now, they haven't reached the equator yet, so that's one thing to be very aware of when you're looking between anaphase and telophase. So if you can still see them as V shapes and they're not really clumped to the end, then you're in anaphase and not telophase. So anaphase is when our centromeres have been separated. Is the picture of the cells what we would... Yes, it would be. I'll show you in a minute what it would actually be. And then you're pulling them apart to the poles. Then you get telophase, and you can see telophase down here. So telophase, this is kind of like a really fuzzy but bulk together, which shows us that this is telophase rather than anaphase. Oh, crumbs, give me a second. It's because I've moved it from one place to another. I just need to format the background because you cannot see the writing. There we go. Right, telophase. Chromatids have reached the poles and are now called chromosomes. Nuclear envelope reforms. Chromosomes start to uncoil nucleus. So these are all things of just telling you about the key things to memorize, basically. So when you're looking at those kinds of descriptions, they're your what you need to memorize. So for me, going back to what I said before about my trigger words, telophase, poles, chromosomes, envelope reforms. That's the sort of thing that I would be using as my trigger words. Telophase, poles, the chromatids have reached the poles, they're now chromosomes. Then you have your nuclear envelope reforms, because we're going to start to actually have it now within its compartment, keeping it safe. The reason there's a nucleus in eukaryotic organism is because as the cell got bigger, as it got more complex, it therefore needed a better way of protecting its DNA from damage. So by housing it in the nucleus, keeps it safe and therefore less likely to be damaged, because the repairing of damaged DNA is energy consuming. That's not a good way of functioning. So they protect it by surrounding it with a nuclear envelope. And then cytokinesis, obviously you've got that splitting, whether it's the animal cell or whether it's the plant cell, in the fact that you've got that cleavage furrow. Basically, these two parts of the membrane need to be brought together so that they can fully separate. A bit like when you do endocytosis, where you have your pseudopodas come out and then they are able to separate. It's the same scenario here. If I bring these two together, they're going to be able to fuse. And with the plant cell, it's slightly different because they have to do the cell wall. And so what they do is they put these vesicles, these Golgi vesicles, in place along the metaphase plate. And what then happens is they start to fuse together whilst the building of the cell wall happens before they separate out. It's really fascinating. And then, Tills, just to finish for you for the mitotic division, if you are given a picture and you have to identify which phase it's in, you will then be told to work out the mitotic index. So as long as you know which one it is. So if I tell you that there are 45 prophase, 8 telophase, 4 metaphase, 8 anaphase, and 325 that are in interphase, you should be able to work out your mitotic index. And you do that by adding together all of the cells that are going through mitosis and then dividing them by the total number of cells. So therefore we'd have our 65 going through mitosis divided by our 390, and we'd be able to work out our mitotic index from that. I hope that's helped, Tilds. I know that was a bit of a long because I was answering other questions. Oh no, don't drop bio. I get why students drop bio because it is tough, and if you don't like it, then it is really hard, and you have to be able to persevere because the content just never stops. especially if you are in year 13 right now. You'll be feeling so overwhelmed because you're learning more and more complex stuff, and it just seems to keep going, keep going. And if you haven't even started module 6 yet, I'm really sorry, but it's still coming and it's tough. um Guys, I will be back on Wednesday as I always do at 9:30 again. Thank you for joining me. Thank you for liking, sharing. Thank you for purchasing my revision guides. Everything you guys do means that I can keep doing what I love, and that is helping you to love biology, even just a little bit, and I feel good. Well, I picked it for a filler subject, and now I'm year 13. Oh, Katie, you picked as a filler subject. Wow, you did not pick well. Sorry, that is rough because it is a tough subject if you don't love it. Katie, that's amazing. That is awesome. Well done. You should be so chuffed to yourself, especially if it's a filler as well. Well done. That is really, really great progress. Now you're on the way to get that A. You should be aiming for the A star. Go for it. Oh, absolutely. So would I be. That's awesome. You should be so proud of yourself because biology is tough. It is really tough. Right, guys, I am going to go though because I need to go to bed. I am going to try and work on Patterns of Inheritance study guide for tomorrow. See how far I get through with that one. So keep watching this space, and I will be back, as I said, at 9 o'clock on Wednesday for a bit more support. So, oh, I'm glad, Ella, you found it helpful. I'm really pleased. Guys, have a good evening, and I will see you. Oh, thank you. See you.